So my name is Clemens Kaminski. Thank you very much for inviting me to talk here today. Just as a brief introduction, so we are technology developers. We develop optical methods, and we try to build tools that allow us to look at biologies on all scales, not quite down to the scale that Radu just talked about. I wish I could do that. But we do everything from single molecules up to organisms, very small organisms, I have to admit. You see here some examples of single molecule imaging of amyloid beta misfolding and growing into fibrils. This is this protein that is associated with Alzheimer's disease. So these are looking at things molecule by molecule, but I'm not talking about this scale. I'm talking today about imaging in living cells, and for that, these single molecule techniques are too slow. So I'm talking particularly today about a technique called structured illumination microscopy and developments we've made in that field, and I will show you how we use it to look at organelles and give you one example. Namely, we look at the dynamics of the ER. And so before I start, I'd like to thank my group. We work very closely with the molecular neuroscience group here in Cambridge, and the work I'm presenting is collaborative with scientists here in Cambridge. So they're mentioned here, and also with Professor Lucas Kapitein from the Netherlands. So very briefly, for those who don't know, I'll just show you how structured illumination works and what we're doing, what the problems are, and then I'll give you some examples of it. So the idea of it is very simple and it's very elegant. The idea is to generate frequency beats by putting structures, illumination structures, onto your sample. And to explain what that means, consider these two objects. You've got on the left a shirt with a slowly varying stripe pattern on it. And on the right, you've got one with a much faster pattern on it, which the stripes are much narrower, perhaps difficult to see for some at the back. You can imagine if your imaging system becomes bad, then these stripes will disappear and you can't resolve them anymore. And so the left shirt has a low spatial frequency pattern, and the right has a high spatial frequency pattern because the lines change quickly as a function of distance across this thing. So in a microscope, these high frequency patterns disappear. And the question is, how can we get that information through the microscope despite the fact that it has limited resolution? And this is the trick of structured illumination microscopy. What you do is you illuminate the sample with a pattern. So the light pattern is itself structured. So if the light pattern shown here on the left has a spatial frequency KEX, and you have your shirt with high spatial frequency, the object that you want to image with a spatial frequency K0, then this happens. And so the illumination causes essentially you get a multiplication of the patterns. And this multiplication causes these low frequency beats to appear, these beat patterns. These beat patterns are called moire fringes. You've all seen them. If you have gardines at home, you sometimes see these large patterns, although you don't see the checkered patterns of the gardines themselves, the laces. You can see those. And so the idea is really to just project patterns and change the orientation of the illumination pattern. So we sample all the spatial frequencies inside the sample, inside the object, and then we use mathematical techniques to reconstruct the object information from these beat patterns. That's the idea of structured illumination microscopy. And so I'm sorry for the equation, it's the only one, it's a very simple equation. So we have an object, it changes, it has spatial frequencies as a function of x or whatever, and you multiply it with the illumination pattern, and that gives you the image with all these funny stripes in it. And so, of course, if I know what my excitation pattern is, And if I know what the image pattern is, then I should be able to work back what the object was. That's the idea. And we can do this very efficiently in Fourier space. We use Fourier techniques to do this. It's a deceptively simple equation. It's actually a very hard thing to do. It's mathematically not a nice problem. It's an inverse problem. And so if you deal with noise, this becomes tricky to do and computationally intensive, but it's doable. So how do we generate the patterns? So the patterns are generated by interfering two light waves. So they're shown here by these blue arrows. Can you see the mouse? Yes, you can. The blue arrows, they come through an objective. So the way we do this is we have an objective, an illumination objective, shown down here. We put two beams, which we focus into the back focal plane of this objective. That creates these two waves coming out, which is shown here in this inset, these blue waves that come out at an angle. And these two plane waves that come out, they interfere in the sample. So the sample sits here on a cover slip, and we get interference fringes on the sample. And that is what generates the pattern that we use to project. So the way we do this is you use a grating. Most often in commercial instruments, you use gratings. Gratings can be electronically produced, but in essence, you have a grating. And so here you have a green beam that's split by the grating, by diffraction, into two beams. And so these two beams are then focused into the back focal plane of the objective. And then this gives you the two waves that come out at the end of the objective and interfere and give you the pattern. The problem with this, so these are commercial designs, usually done this way. The problem with this is that this works only for one wavelength. So the grating has to be optimized for each wavelength. So for example, here the red, if I do this with the red color would here miss the objective, right? So that's no good. So it's optimal only for one wavelength. The other problem is that you need to, of course, rotate the grating to get all the different spatial frequencies in the samples. We need to project lots of patterns. In fact, we need to do 9 patterns. We need to do 3 orientations. And we need to do 3 phase shifts for each orientation. So each pattern is stepped 3 times in each orientation. And so it's 9 images to get one normal image. And so if we want to image several things at once, that's difficult, right? Because we have to do this sequentially. We have to sequentially change gratings, rotate, and do this kind of stuff. So it's not so easy to do. So a better way would be to actually use a physical beam splitter. So you split the beams with a prism like that, for example. And so you align the incoming beams onto the microscope lens like that. And so in this case, this works for any color. We can put any color in simultaneously. The only problem is, how do I rotate this? How do I produce the phase steps and all this kind of stuff? And so this is where we came up with this idea. So this is the heart of the instrument that we've developed. So essentially, we use what's called a microsonic interferometer to generate these patterns, to split the beam into 2 parts. So physicists will here recognize the microsonic interferometer. So here's the beam splitter, that big cube. The laser beam comes in, and then is split into one beam this direction, and another beam in that direction. And then retroreflected, and that creates my 2 beams coming back. And they are then going towards the microscope and then interfere in the microscope. And in fact, if you look carefully, so you see 3 sets of mirror pairs. So this mirror here corresponds to a mirror here, that middle mirror to the middle mirror here, and that one there. And so what this does is each of these mirrors pairs are adjusted in such a way that we can rotate the patterns. So the beams are rotated by addressing different mirror pairs. And the other thing is that by sweeping across each individual mirror, we can actually also do the phase shifting very quickly. And the beauty of this is we have one moving element to achieve all this: pattern rotation for any color you like, and pattern shifting for any color you like. There's a galvo mirror sitting here, which we can spin very, very quickly, so we can quickly address the different mirrors. Here's an animation of this, if it works. So you see the incoming beam, it bounces off the mirror here, it goes onto the beam split, and you can see how we can address the different mirror pairs by just rotating that mirror. And so the beams come back, interfere, and produce the patterns. It's really a nice thing because it's relatively simple to set up. Everybody can set this up. It doesn't cost very much to do, and it actually ends up being a much better microscope than the ones that you can buy. It works with very large fields of views, and it's also achromatic. It works for any color you like. There is a small problem with this, and that is if for people who use SIM for structural illumination microscopy, in contrast to the grating approach, we have one difference, and that is we cannot control the phase stepping as accurately as we can do with the grating approach. So that means for different colors, the phase steps are not equidistant. So I do image 1, 2, 3, like that, image 1, 2, 3, like that, image 1, 2, 3. The steps between those are not quite the same for all the colors. And that means that this classical Fourier reconstruction that I mentioned earlier, that this doesn't work. The classical ways of reconstruction don't work. So what we did is we used a different approach. We used machine learning to do the following. We essentially mimicked what different objects would look like if we imaged them through our microscope. That's what we modeled physically. So we took random images, large libraries of images from the web. This is a famous data set for vision, for computer vision. It's called the diff2k data set. We pretended these were objects we would image in the microscope. So we just made them into black and white images, made them to look like they might look as if they were imaged through our microscope. Then we multiplied these images with these patterns we generated in our microscope. We convolved them with the resolution of our objectives and functions, the resolution of the microscopes that we use. And then we add noise, and so this is like pretending what the raw data might be that comes from our microscope. And then we had a machine learning algorithm try and work back what the ground truth image was. And because we have thousands and thousands of images available to us with infinite resolution, so to speak, we can actually make it a very, very accurate predictor for almost any instruments you want. It's not actually limited to our type of SIM imaging. You can use this for any SIM machine and get very, very nice reconstructions that way. That if you're interested, it's available. You can actually just drop data in on this website, just drop it there, it will reconstruct it for you. You can train it. The software is all freely available. It's now becoming very popular for microscopists to use. Here's some performance characteristics. It does really, really well. So on the right-hand side, you see the output from simulated images from our method. In the presence of noise, it performs really well. And even on real data, if you compare it to existing sort of state-of-the-art methods, it does a really an almost better job. It does a better job when you have noise in the image. And so I don't want to spend too much time on this, but what is an advantage is that once you've trained the algorithm, which takes about a day or so to do, you put your system parameters into the model, you use these artificial image data sets, you get a training data set, which is based on a real physical model. It's then optimized, the algorithm, for your system, and then you reconstruct whatever you feed it from your microscope, and then the reconstruction is incredibly quick. So to reconstruct images with this takes a few milliseconds on a graphics computer. And then you can do almost real-time imaging with this. So what do we do with these things? I'll show you two applications. So we're now able very quickly to image things that go on inside the cell. And so we looked at the ER. And so you can now look in live cells at the ER network and see how it's dynamically changing. You see the sheet and tubule structure in there. And actually looking at this together with our colleague Edward Avezov, we noticed, if we looked a bit more carefully in a bit more detail at this, that these ER networks, these tubal networks, they display these pinching points. So if I turn on the microscope here, you can see every now and then you see that you get these pinching movements on these tubules. And this was really puzzling to us. And I don't know if you can see it here too. Hang on. Yeah. So this was not known before, right? And you see this all over the network, in fact, in lots of different cell types. And what's more interesting is that this pinching is ATP dependent, it's energy dependent, right? So if you deplete ATP in cells, you can see that the frequency of these pinching events goes down. So it's clearly an actively controlled process. And we've also combined this method with single particle tracking, single molecule tracking. So we used luminal proteins from the ER, tracked them with a tag, and then measured their velocities inside the ER lumen. And you could see that the flow velocity in the lumen correlates with this depletion in the same way. And so there's a plausible link, right, between these pinching things that we think of as like a peristaltic pump in some way. So you have a sausage which you squeeze and then you get flow from each side. So it randomizes the flow inside this thing. So it's an ongoing project, and we don't really know what the origin is of these pinching movements. So what the motor is that does that behind it. There are lots of theories out there, but it just shows you that looking at things with good resolution inside cells, you can see new things. Another thing where we now use this two-color or multiple-color possibility with structural illumination microscopy with our system is looking at several organelles at once and looking at their correlated motion. So here you see lysosomes inside cells, and you see the ER network whizzing around in there. And so what is interesting is that you see very often a highly correlated motion between those two things. I mean, it's not completely surprising because there are lots of contact points between lysosomes and ER. But if you look at some of these individual events here, you can see that sometimes these lysosomes seem to drag behind them, ER tubuli. And they seem to sort of almost like sewing machines to bring the tubal network to make connections inside. So we can see down here, for example, here you see, I don't know whether you can see my mouse still. Let me try this here. Yes, I don't know whether you can see this. Down here, you see how this lysosome, the green bit, is drawing the ER down and making this connection here. And so this led us to speculate that, in fact, perhaps the lysosomes actively control the dynamic topology of the ER. And to test this, we introduced it's a well-known, we used a construct which has a mutation where actually so we used a protein here, VAPA, which is known to be providing an anchor point for lysosomes onto the ER. And using a mutation in there, you actually reduce the ability of that anchor, right? So to bind the lysosomes. And so when you do that, you see, in fact, frequently events like this. Let me see if I turn this off again. I don't know how to do that. Oh, yeah, here we are. So you see, for example, here a lysosome is sort of pulling up a tubule, but it detaches, and immediately the ER tubule retracts at very high speed. And also the other thing that you see is actually the lysosome speeds up as it releases the ER tubule, and that is because the resistance, the pull resistance, is actually reduced. So it continues along its track, pulling the ER behind it, and then when it loses that capacity, you see the ER retracts. And so if you look at, if you analyze images like that, so you see that the length of tubules made inside the ER correlates a lot with whether at the end of that tubule there's a lysosome present, pulling it, which is this green bar. And all of these make connections with the other network. The ones that don't have those things, they make much fewer connections, many fewer connections, and in fact, many fail to make any connections. These are the red ones here, in this case. You can do this also in more complex models. Sort of, one of our students, Francesca van Tartwijk, used a frog model. So these are retinal ganglion cells from frogs. So you have an explanted eye, and the RGCs, the retinal ganglion cells, grow out of the eye. And you can see, first of all, if you have this mutation present inside this map mutation present inside this, the RGCs don't grow as nicely. So you can see that here. So this has a developmental effect. And the other thing that you see frequently, you see events like this. You see here, oops, can't now turn off the laser pointer. I don't know how to do that. Yeah, there we go. So you see here, in magenta, you see an ER structure going through the axon of one of these retinal ganglion cells inside this model. You see a lysosome coming along with a bit of ER wrapped around and reconnecting a broken piece of ER. So you can see that mechanism has plausibly a connection to maintaining ER health. To test this mechanistic, this actually the lysomes being the active driver of ER topology, dynamic topology. So we collaborated with Lucas Capitaine, and actually we used optogenetics to control with light how we connect lysosomes to kinesins. So we made an active anchor which only forms when you put light onto the construct. And so as soon as that happens, as soon as you do that, the kinesin starts moving the lysosomes from the nucleus here out to the periphery. And so what you see here in magenta, so in green, you see the lysosomes. They're sort of located around the nucleus quite a bit of this particular cell. In magenta, you see the ER network, and we used an algorithm that we have available to skeletonize the ER and make it visible. So this is a reconstruction from images, but it's easy to see this. And as soon as I put the light pulse on, the motors will drive these lysosomes out towards the cell periphery, and you will see how the ER network reconfigures as a result of this. So if I do this, so light pulse comes, you see they're all moving out towards the cell periphery. You see massive rearrangements around in the ER. And in fact, where there are no lysosomes, nothing happens. So you can see this active role that these organelles take. And I'm running out of time, so we've also looked at this. If you do metabolic stimuli to the cell, so it's known that lysosomes reposition themselves in response to metabolic signals or stress. And so as soon as they repositions, you can also see how the ER also repositions as a result of that. And so with that, I finish by just giving you some very superficial summary. So the way structured illumination microscopy works is by generating frequency beats. and you use these machine learning methods to actually reconstruct very quickly in multiple colors. And these methods, which are definitely very, very intermediate on the super resolution scale, they allow you to find out new things in living cells. Thank you.
Very much.
Thank you for nice talk. If you compared custom your sim imaging and commercialized sim imaging, is there any difference between resolution?
No, the resolution, if you optimize a commercial sim system, can be very good. For one color, it's very good. Ours is certainly better if you have several colors imaging at once. So we can really put all the colors in at the same time and get all the patterns simultaneously. So that will not be possible. We can get to just about under 100 nanometers, wavelengths dependent.
So you said the resolution goes down to about 100 nanometers. Would that allow you to sort of track viruses going through a cell, for example?
Yes.
Have you ever tried that?
You can't resolve them in the sense that Adrian just said, Rado just said, but you can certainly track them easily. That's not a problem. We've done that. In fact, we've used other methods in combination with expansion microscopy, looking at virus assembly inside cells.
Really interesting talk, and I loved the lysozyme sort of remodeling of the ER network. That was amazing. I think in some of the videos you showed the timescales were seconds. Was that right? And I wondered, could you say something about, I mean, how long can you take these movies for, and how do the dynamics that you're monitoring compare to other techniques that look at things like this network rearrangement? Thank you.
So we can image with multiple colors at maybe 25 hertz, 20, 25 hertz. We're limited, in fact, by the camera technology at the moment. This trick goes quickly, but then you need to also put a lot of light onto the samples, and then the question is whether the samples like that. So the light dosage is a really important thing, and that's where then these algorithms come in. So we try and put the light dosage down, get lower quality images, but perform better reconstruction. So it's essentially exactly that game we have to play: resolution, speed, quality of the images, survival.
I was intrigued by that peristaltic pump that you mentioned. So that is like process, presumably fueled by ATP consumption, right?
Yes.
So there must be some ATPases associated.
Yes, I don't know what they are,
Yeah.
but it would be really, really interesting to start and search what goes on. You know, presumably something around there.
Yeah.
There are obviously also many membrane reshaping proteins or ER shaping proteins, but they, you know, it's very hard to think about something that's quick enough, right, to do these very, very fast processes.
And the objective would be the objective of the cell would be to regulate the flow, you think, inside the ER.
I mean, so there is a known conundrum if you think about a giraffe, let's say, you know, which has very long neurons, right, that go from the head to the foot. So, and there's no known active machinery inside the ER lumen that transports molecules around. So if you have a molecule made at a particular point, if you just rely on diffusion for molecules to arrive at a distal part of the cell, it would take days, right? In the case of the giraffe, weeks for that to happen. So we can definitely correlate. We've done some modeling about this. Not me personally. David Holtzman in Paris has done on our data done modeling, and he sees definitely this mixing improves definitely the speed at which the solutes can go through the ER. But we don't have the full story behind us yet.
There is one more question. Yeah.
To follow up from your question, Maria. So if you have these like pinching movements, do you see any evidence they proceed in motion, in sequence, or is it just apparently random?
This is no, so between different locations, it's, as far as we can tell, it's random. So it's a random sort of mixing enhancer that will actually put molecules through more quickly.
It would be like pushing them through.
But I mean, we haven't seen that at all. I mean, but I don't know whether we just didn't see it because we weren't. We were definitely looking, but we might, because it's actually hard to see these things, right?
You will need a giraffe one to follow. Okay, good. So before we thank all the speakers, I would like to ask the speakers to stay here for a moment. So we will take a photo of all the speakers, okay? Yeah, okay, just outside, please. Wait just outside so we know. Oh, it's the other way. People should stay.
Yeah, we stay.
Okay.
Everybody's just there.
Sorry. Everyone stay behind for a moment. You can make a photo, and then you can come out, okay? We promise we don't eat your lunch. Thank you very much. Let's thank the speaker.